बिस्मिल्लाहिर्रहमानिर्रहीम अस्सलामु अलैकुम वरहमतुल्लाहि वबरकातुहू। आज हम प्रोटीन सिंथेसिस का पार्ट टू जो है स्टडी करेंगे। और पार्ट टू में क्या शामिल है? पार्ट टू में शामिल है प्रोकैरियोटिक ट्रांसक्रिप्शन। आप बताइए, हमारे पास एक टॉपिक मौजूद है ट्रांसक्रिप्शन एंड ट्रांसलेशन। सो इसमें ये लिखा था जो है ये आप देख सकते हैं, इसके 3 ऑथर्स हैं और इसको हमने स्टार्ट किया था लास्ट टाइम। तो इसका हमने इंट्रोडक्शन पढ़ा था। आज हम जो है वो डिटेल में पढ़ेंगे प्रोकैरियोटिक ट्रांसक्रिप्शन। स्टार्ट जिसे कहते हैं इनिशिएशन ऑफ ट्रांसक्रिप्शन, इनिशिएशन ऑफ प्रोकैरियोटिक ट्रांसक्रिप्शन। It needs a particular DNA sequence called as promoter in prokaryotes to find the right starting point of RNA polymerase enzyme transcription. It needs a particular DNA sequence. So a particular DNA sequence is required, and this particular DNA sequence is known as promoter. It is present in prokaryotes, and it is required to find the right, precise starting point of RNA polymerase enzyme transcription. This section is located close to the start of transcription. When we see so in detail, so we find that this section, this specific particular DNA sequence known as promoter is located close to the start of transcription. Promoters are important part of the regulatory region of a gene. Promoters are important part of the regulatory region of a gene. So here it is written that promoters are important part of the regulatory region of a gene. In transcription start site, there is generally a purine base and pyrimidine bases which are located on both sides. So when we talk about transcription start site, means we are talking about DNA sequence. So there is generally a purine base, guanine or adenine, and there might be pyrimidine bases, cytosine or thymine bases, which are located on both sides. So these are located on both sides. There might be this sequence CAT, CGT, so on. The synthesis of RNA transcript starts 5' of the promoter and continues towards 3'. So the synthesis of RNA uh, proceeds. The synthesis of RNA transcription is from 5' of the promoter towards the 3' region of the strand, DNA strand. Typically, 5' is written on the left and 3' is written on the right. Traditionally, typically, customary, 5' is written on the left and 3' is written on the right. Because the promoter is near the end of the gene where transcription is initiated, in other words, being in 5' of the gene, promoter region is also referred to as regulator 5. In figure 1b, it is shown promoter region of 7 different genes in the E. coli genome. So here you can see these are 1, 2, 3, 4, 5, 6, 7 uh, regions of E. coli genes where promoters are being shown. Although there is a same RNA polymerase, there is same RNA polymerase, different genes have promoter sequences. Different genes have different promoter sequences. It is not surprising for the similarities between promoters. It is not surprising for the similarities between promoters, especially great similarities of two regions are seen in almost all cases. Especially, we find great similarities of two regions in almost all cases. These regions are known as minus 35 and minus 10 regions. These regions are known as minus 35 and minus 10 regions. The reason is that they are located respectively 35 and 10 base pairs. So it is according to their location. These positions are separated from each other by 15 to 17 base pairs. These positions are separated from each other by 15 to 17 base pairs. Minus 35 and minus 10 regions of different genes do not need to be the same. When we talk about regions, so minus 35 and minus 10 regions of different genes do not need to be same. However, it is possible to find a nucleotide sequence which is compatible with most sequence. This sequence is called as a consensus sequence. So there is present a consensus sequence. The two sequence are not necessary to be same, but they may be compatible with most sequence. So this sequence is called as a consensus sequence. RNA polymerase holoenzyme binds to DNA from this point. RNA polymerase holoenzyme binds to DNA from this point. After that, what happens after that? The double helix DNA is opened and a synthesis of RNA molecule is started. The double helix DNA is opened and a synthesis of RNA molecule is started. The first transcribed base always determines the initiation site in the same position. The first transcribed base the first transcribed base always determines the initiation site in the same position. Transcription begins before protein encoding gene segment. Transcription begins before protein encoding gene segment. Thus, a transcription has five untranslated region. So there is five untranslated region present before because transcription begins before protein encoding gene segment. Bacterial RNA polymerase enzyme is also called as RNA polymerase holoenzyme. Bacterial RNA polymerase enzyme is also called as uh, holoenzyme. This is known as a holoenzyme. Why? This enzyme scans DNA for a promoter sequence. So this is its functioning, that it scans DNA for a promoter sequence. This multi-subunit enzyme complex consists of the four subunits of the basic enzyme. So there are four subunits of the basic enzyme: alpha subunit, beta subunit, beta prime subunit, and omega subunit. So there is alpha subunit, beta subunit, beta prime subunit, and omega subunit, and sigma factor also called as a subunit. So here we see when we see here it in detail. So we find the enzyme scans DNA for a promoter sequence. This multi-subunit enzyme complex consists of the four subunits of the basic enzymes: alpha subunit, beta prime subunit, and omega subunit. And number five, there is a sigma factor, also called as a subunit. In holoenzyme, there are two alpha units and one from each units of the other four units, like the alpha, beta prime, omega, and sigma factor. The alpha subunit is encoded by gene RPOA. The alpha subunit is encoded by a gene RPOA. It is required for association of basic complex, but there is unclear whether it has a transcriptional activity or not. It is considered that the beta subunit encoded by gene RPOB is the catalytic subunit of the RNA polymerase enzyme. So it is found, it is considered It is considered that the beta subunit encoded by the gene RPOB is the catalytic subunit of the RNA polymerase enzyme. Furthermore, beta prime subunit is encoded by gene RPOC. It is known that this unit is responsible for binding to DNA. Sigma subunit is connected to minus 35 and minus 10 region so that it is positioned correct start site to initiate holoenzyme transcription. So Sigma subunit is responsible for correct initiation of holoenzyme transcription. Sigma subunit has also a role to separate DNA strands around 10 region. It has also a role to separate DNA strands around 10 region. In this way, the basic enzyme binds tightly to DNA in preparation process for RNA synthesis. So the basic enzyme binds tightly to DNA in preparation process for RNA synthesis, while the basic enzyme is bound after initiation, transcription starts, and sigma subunit is separated from the rest of the complex. So basic enzyme is bound after initiation, transcription has begun, and alpha subunit is now separated from the rest of the complex. As in many other bacteria, E. coli has many different sigma factors. One of them is sigma 70, its mass 70 kilodaltons. This primary sigma factor is used in the early stages of great majority of E. coli genes transcription. So one of them is sigma 70, and this sigma 70 factor is used in the early stages of great majority of E. coli gene transcription. Other sigma factors recognize different promoter sequences, thus facilitated different factors. The same basic enzyme recognizes different promoter sequences and transcribes different groups of genes. Other sigma factors are there to recognize different promoter sequences, thus facilitated different factors. The same basic enzyme recognizes different promoter sequences and transcribes different groups of genes. Now comes elongation. When RNA polymerase moves along the DNA strand, enzyme wraps the upstream transcribed DNA helix back while opening the downstream DNA helix. So now elongation initiation has started, and now the elongation is taking place. So RNA polymerase enzyme is moving along the DNA strand, and the enzyme is wrapping the upstream transcribed DNA helix back while opening the downstream DNA coming next. In this way, enzyme holds a portion of single-stranded DNA region. This region is called transcription bubble. This region is known as a transcription bubble. Polymerase enzyme in bubble watches the binding of free ribonucleoside triphosphate to the next open base and DNA template. Polymerase enzyme is present in bubble. It recognizes the binding of free ribonucleoside triphosphates to the next open base and DNA template. And if a complementary match is made, that base is added to the messenger RNA chain. And at the end it comes termination. A single gene transcription is terminated out of the protein coding segment of the gene at the end of the 3' untranscribed region. So uh, a single gene transcription is terminated at the end of the 3' untranscribed region, uh, 3 UTR. For the termination of RNA polymerase chain, the elongation in transcription continues until it recognizes a specific nucleotide sequence acting as a signal. This process continues until it recognizes a specific nucleotide sequence that is acting as a signal. The signal initiates the oscillations of RNA newly formed with its nucleotide and template from the enzyme. So it starts oscillations of RNA formed in prokaryotes, for example, E. coli and most bacteria, in termination step of transcription. This signal can be from two different mechanisms. These are row independent mechanism, the real mechanism, and RHO row dependent mechanism. So first there is row independent termination, that is real mechanism. Row independent terminator is the easier one when we compare the two systems. It is the easier one when we compare the two systems. As a result of this mechanism is called as a simple termination. This mechanism is called as a simple termination. Row independent terminator sequence contains about 40 base pairs. Row independent terminator sequence contains about 40 base pairs. These bases are terminated rich in GC base pairs. G and C in the template sequence will respectively give C and G in transcription, and RNA will be rich for GC in this region. These cytosine and guanine bases being complementary between them can form hydrogen bonds and end with the hairpin loops GC base pair is more stable compared to AT base pairs because GC is connected with triple bonds. There are three hydrogen bonds between G and C as compared to AT in which there are two hydrogen bonds, so it is more stable. While the AT or AU is connected by two hydrogen bonds, hairpin loops consisting of GC base pairs are more stable than loops consisting of AT base pairs. So hairpin loops consisting of GC base pairs are more stable than loops consisting of AT base pairs. Cycle follows with 8 U base in accordance with the A residue that is on the DNA template. So this cycle follows 8 U bases in accordance with the A residue that is in the DNA template because both there should be a complementary sequence. In this mechanism, it is believed that the polymerase enzyme stops after U bases are synthesized. So base pairings that consisted with correspondence of A bases in template DNA stand to U residues. Weakens the combination of DNA RNA. This facilitates the removal of RNA chain from enzyme complex and template DNA chain. After this, there is row dependent termination. While RNA polymerase enzyme can terminate RNA transcription from the hairpin structure itself, sometimes termination regions may not create hairpin structure. Sometimes hairpin structure may not be formed. In this case, it is required row-dependent termination mechanism. In this mechanism, it needs to have row cofactor for recognition of the termination signal by RNA polymerase enzyme. Here, in this case, a row factor is required for recognition of the termination signal by RNA polymerase enzyme. With the row-dependent termination signals, RNAs do not generally have U residue sequences and hairpin loops. They do not have U residue sequences and hairpin loops. Instead, in this row-dependent termination, instead they have 40 to 60 nucleotide sequences, including an upper portion called as row Blandly region, which is rich in C residue but poor for G residues. Uh, when Rho binds to a single-stranded RNA, it is a hexameric protein that can hydrolyze ATP. So when Rho binds uh, to a single-stranded RNA, it is a hexameric protein that can hydrolyze ATP. Consisting of six similar subunits, Rho binds to the Rho binding regions on the new RNA chain. After binding, Rho facilitates the oscillations of RNA from RNA polymerase. These regions are located upstream of sequence where RNA polymerase enzyme tends to stop. So today we studied prokaryotic transcription, and inshallah next time we will study eukaryotic transcription. So you can you can put your questions in group or in a comment section, and we will try to answer these questions. Thank you. Allah Hafiz.